going to continue on about this idea of membrane transport and membrane function. Shown here is a manometer tube, or this U-shaped tube, and the little dashed line that you see in the middle represents a biological membrane that is selectively permeable. You can clearly see with the red dots on the left side that there is a concentration gradient for these solutes. Very highly concentrated on the left, with none of these red dots on the right. Now, if I told you that these red solutes were permeable across the biological membrane, what would you think they would do? Clearly, they would diffuse. They would spread out from a high concentration down to a low. They would continue to do this until they reached equilibrium, which means that there is no net gain or loss of the solutes across the membrane. It does not mean that they stop moving. They can still move from one side to the other, but there is equal transfer of one solute from one side to the next. So in the end, we would reach equilibrium, and the solutes would look like this. But that is not always the case. Solutes don't always have permeability. And we learned earlier in the segment that we do have sidedness to the cell membrane. We have more sodium on one side, more potassium on the other side, and these gradients are being maintained. So if the solutes cannot move, and we find solutes of unequal quantity per unit of volume, what will move? And the answer is water. There are two reasons why water moves. Water moves because of a hydrostatic pressure or an osmotic pressure. And this movement is always passive. There is no chemical energy required like ATP, and it's unsaturable, meaning it can continuously move. There are pores in the biological membrane that allow water to move through the cell membrane. These are called aquaporins. Aqua means water, and a pore means it's a pore or a tunnel, if you will, through the cell membrane. And there are many different types, and do not worry about aquaporin type 1 versus aquaporin type 2. Let's start with the bottom bullet where it says hydrostatic pressure. This really is a pushing pressure. Hydrostatic pressure, the best example of that would be your blood pressure. We put the blood pressure cuff around your arm. We inflate the cuff to occlude blood flow through your brachial artery. And then we release the cuff pressure until your blood pressure is higher than the cuff pressure, and we start to see the dial move or kind of thump around. And in our stethoscope, we would hear some turbulent blood flow. So hydrostatic pressure is an outward pressure, a pushing pressure on the vessels in your body. This allows, or it is the driving force that causes plasma water through the capillary clefts and into your interstitial space. This pressure is what causes plasma to deliver nutrients to your tissues and pick up waste products. If you were to pick up your water bottle, as shown here to the right, and pushed on the water bottle, clearly you would get the water inside to move superiorly towards the neck of the bottle and even come out of the bottle. That's a hydrostatic pressure. Even the water just sitting in your container is exerting an outward or pushing pressure on the bottle, and the water takes the form of the bottle itself. This is very different than osmotic pressure. Osmotic pressure is a pulling force, where solutes that are found in unequal quantity across a biological membrane per unit of volume will pull water towards them. We say particles suck. It's actually easier for physiologists to measure the solute concentration, more solute means less water in that solution, than the actual water itself. But osmosis is movement of water based on a water concentration across a cell membrane, the difference of water concentration. This osmotic pressure is the driving force for water movement across cell membranes. That's in striking contrast to the water moving in plasma to the interstitial space across capillary walls. So let's repeat that experiment again. Here's our manometer tube. We see the red solutes in unequal concentration on the left side. And if now I tell you that the cell membrane is impermeable to these solutes, what would happen? And my picture is showing you the distribution of water. We see on the right side there was water volume loss because the water moved to the left side in an effort to dilute those solutes. So this water shifting, this net movement of water called osmosis, is when the solutes are impermeable, there is unequal concentration per unit of volume, and therefore the water will move. And water will always move across biological membranes. And that is the reason why we need to be mindful of this with our body fluid compartments. And I'm going to show you real examples of how this shifting can occur. This is basically the basis for how laxatives and diuretics work. We will learn more about laxatives and diuretics as we move on throughout the semester. Until then, understand that a diuretic or a laxative is something that cannot move across a biological membrane. It's a solute that is impermeable. And therefore, if there are unequal quantities of these solutes on one side of a cell membrane than the other, then the water will shift towards the side of these chemicals. And again, that is the basis of how these work. They are disrupting body fluid compartments. Let's focus on osmotic pressure a little bit more because it is so important, and you will do a PhysioX experiment with this. Here's our manometer tube again. And on the left side again, you're seeing a non-diffusible solute. That means it's impermeable. On the right side, you're seeing pure water. Scientists don't really have a direct way of measuring osmotic pressure. Instead, they use a device, this piston that you can see here in black, on top of the left side of the tube. And this piston is pushing. It's a pushing pressure. That's a hydrostatic pressure. And what they do is they allow the water from the right side to diffuse or try to diffuse to the left side. And of course the volume on the left side, or side A, would go up. But if I were to be pushing down on this black piston with equal but opposite force, the water level would not rise. And I can actually quantify in different units of measure, like pounds per square inch, of how much pressure I have to apply to that piston to prohibit water from shifting from the right side to the left. This is exactly the experiment you will do in your simulation exercise in PhysioX. If we say something is isoosmotic, it means it has the same osmolality as our body fluids. The reference point is always the solution inside the cell, the intracellular fluid. That should always be about 300 milliosmolal. If we were to take a cell from your cheek and put it in a beaker surrounded by solution that is isoosmotic, we would not expect to see gross shifting of water from one side of the cell to the other. If I were to put the same cheek cell into a beaker of fluid that is hyperosmotic, that means the osmolality is higher now in the beaker compared to within the cell. Again, the starting reference point is always inside the cell. If I were to put your cheek cell, your squamous cell, in a beaker that is hypoosmotic, that means the solution in the beaker has a lower osmolality compared to the intracellular osmolality of your cheek cell. And we would start to predict or ask questions if water would shift from one side to the next. Osmotic pressure is attributed to the osmolality of a solution. Osmosis occurs when water moves from a solution with fewer particles to one where there are more particles per unit volume. The particles in this case are impermeable. They cannot diffuse. And therefore water will, trying to reach an equal concentration on either side of the membrane. You can always remember this as particle suck. The side that has the higher osmolality, when the particles cannot diffuse, will have more water drawn to it. Here's a real-life example. Let's take into consideration diabetes mellitus. Over 20 million people in the United States, and this number is increasing by the year, have diabetes mellitus, whether it be type 1, which is pancreatic failure, or type 2, which is obesity related. Both of these types of diabetes on a molecular level have a problem with membrane transport. That is the problem with diabetes. This lack of membrane transport leads to a problem with glucose levels in the blood. They increase. Because the cells of the body, shown here on the left side of the picture, the cell shown here, cannot insert glucose transporters, they cannot take the glucose from the bloodstream and internalize it. Therefore, the blood glucose levels remain elevated. We now have a disturbance in our body fluid compartment osmolalities. You can see here all the S's that I've shown here as glucose for sugar within our blood vessel. The plasma osmolality is no longer 300, but instead 304. But adjacent tissues near this blood vessel are still at 300. So clearly the plasma, an extracellular fluid compartment, has a higher particle count per unit volume than the intracellular compartment. And that means the osmotic pressure is higher now in the plasma. This means the surrounding cells will lose water to the plasma through osmosis, and the person's cells will crenate or shrivel. That means they're becoming dehydrated. This is a problem. To exacerbate this dehydration process, because they have more water now going into their plasma, they will have a higher hydrostatic pressure. And now with this higher hydrostatic pressure, their kidneys will filter more plasma. That means more urine production. That's polyuria. And that's going to exacerbate their dehydration. High blood osmolality, leading to more fluid loss from surrounding cells, leading to more urine production. The person's brain, specifically their hypothalamus, will detect this problem, will detect the high blood osmolality, and this will increase their thirst drive. The person will have increased thirst and will be seeking out more water. And it's not a winning battle, it's a losing battle. They will continue to be dehydrated. This increased thirst is called polydipsia. Because the cells are without their proper nutrition, their go-to fuel source is always glucose. They are going to be starving in the face of plenty. This will also instigate another behavior through the hypothalamus. The person will have increased hunger, and they will be eating voraciously. But it's, again, not a winning battle. They're not able to get glucose into their insulin-dependent cells. This increased eating behavior is called polyphagia.